Good afternoon, everyone. Welcome to the Wednesday afternoon lecture, a special lecture this time, the G. Burroughs Miter Lecture. This is an annual event in honor of Dr. Miter, who was the first NIH Director of Laboratories and Clinics. And so we give, and you can read more about him on the back of the sheet from today. This is always a special opportunity to recognize an intramural scientist who has made outstanding contributions to biomedical research. And I think this year we've done very well in choosing Lou Staudt as the Miter Lecturer, given his remarkable contributions to cancer research, particularly in the areas of lymphoma, utilizing tools of functional and structural genomics. And it's a pleasure for me to be able to introduce somebody that I've had the privilege of knowing for quite some time and learning from in terms of the way in which he has really been a lead in this whole idea of precision medicine before it started getting thrown around so loosely by so many people. Lou was doing it by figuring out how to take apart the mysteries of different types of lymphoma, much to the advantage of patients that he has helped by his discoveries in the way in which they've pointed to specific therapies that are appropriate for subgroups that were not apparent just by looking through the microscope at what those cells look like. Lou got his undergraduate degree at Harvard College, went on to get a MD PhD at the University of Pennsylvania, focused in immunology. After a postdoc a year at the Wistar, he ended up in a very interesting place, namely the Whitehead Institute in the laboratory of David Baltimore, where he carried out a postdoctoral fellowship. And I've just learned that the person at the adjacent bench was none other than Mike Leonardo, so it's quite a nice connection here between the Whitehead in the 80s and the NIH in the 2010s, which is, I guess, where we are. He came to NIH in 1988 as a senior staff fellow, and as you can see and will hear in his presentation, took advantage of this remarkable facility and all the capabilities that happen here in intramural NIH, bringing to that his own exceptional intelligence and creativity and vision to produce an incredible series of presentations and discoveries about cancer, and now serves as the co-chief of the lymphoid malignancies branch and the director of the Center for Cancer Genomics, as well as being chief of the lymphoid malignancies section in the metabolism branch. He is an elected member of the National Academy of Sciences, and in 2009 received the Damashek Prize from the American Society of Hematology. So a distinguished scientist in our midst. We're fortunate to have him here this afternoon. Please join me in welcoming Dr. Lou Staudt.
Thank you. Well, as it should be obvious, it was not clear to me I'd have the privilege of being introduced by Francis today, but we are very glad he's here to do many things for us. And more generally, I think you'll see that really my career would not have been possible without the Human Genome Project. I was sort of living through it and inspired by it, and I just used it throughout. And I'm going to try to tell you some sort of an arc of, I'm going to do a little bit of history, but there's going to be a fair amount of new data in this lecture as well. But the important thing and the special thing I think about, one of the special things about being here anyway, is our ability to seamlessly think about disease processes, work on them in the laboratory, and then try some things out and hopefully help some people along the way. And so that's what I've tried to do, and I'll give you some examples of ping-ponging between the lab and the clinic throughout. And as Francis said, there was not a name of what I was working on at the beginning, but it's now got a name, Precision Medicine, and I'm embracing that. I want us all to go in that direction. And this is an illustration of why. So in after I got my feet on the ground as pretty much a basic scientist in developmental immunology, when I came here, it dawned on me that they did recruit me to the Cancer Institute, and what was this problem of cancer, and so I said, I know about B lymphocytes, so what are the cancers of a B lymphocyte? And they were lymphomas. At least I had learned that in medical school, but I hadn't learned, you don't learn much about cancer in medical school, but basically opened up the authoritative textbook on lymphomas and at the time, and this is what I found. Now, I'm illustrating this first. What this microscope, I have to say, is near and dear to my heart. This is my dad's microscope in medical school. He gave it to me when I was a young lad, and I looked at a lot of pond scum in this thing and other things to pass the time. And honestly, how lymphoma was diagnosed when he went to medical school was how it was being diagnosed in the mid-90s. People were looking under the microscope and they said, gee, the cells are large and they're diffuse in their distribution in this tissue, so we'll call this diffuse large B-cell lymphoma. And so that seemed to not have a molecule in sight. I was a dyed-to-the-wool molecular biologist at that point and thought there might be some opportunities. So fast forward to 2015, and it's sort of remarkable that in those ensuing maybe 15 to 20 years, this is a figure taken, a cartoon from one of my papers from my lab last year in some incredible molecular detail. It's probably not all right, but it's a good model of where we are, and you can see a lot of proteins and their interactions and ubiquitin modifications of those proteins, and that's what we think lymphoma is. It's not these cells under the microscope, and it is this kind of molecular understanding that leads us to have an idea about therapy. You don't get an idea about therapy it seems, by looking at the shape of the cells, but this gives you some good ones. And so then, as I mentioned, I was growing up in science as the Human Genome Project was developing. Now, you guys are just going to the internet, all of you just typing in your gene, learning every exon. We didn't at all know this. We fortunately knew the structure of the human genome. We didn't know where the genes are. We didn't know how many there were. We didn't know how they were expressed. We didn't know squat, really. So then we started early with I say from the Museum of Antique Genomics. This technique is not used too much at all, but it's a cDNA microarray, and every spot here was a lovingly PCR'd spot of cDNA in my own lab, 18,000 here. The first incarnation of this was done in collaboration with a great scientist, Pat Brown at Stanford, and then we brought it here. And you can see the penny is 1999. So this was when it was all breaking for us, and we turned our attention to use this gene expression tool to look at expression differences in lymphoma samples. And without belaboring it, we very easily found that there were two large clusters of samples that were associated with different gene expression profiles, and we called one, for reasons that will become clear, the activated B-cell-like or ABC type of diffuse lymphoma, and one, the germinal center B-cell-like or GCB diffuse lymphoma. And actually now I'm happy to say this is part of the World Health Organization classification of lymphomas, and I'll tell you why. It's not just based on some gene expression. You can always find gene expression differences. The initial clue was that there was also a difference in survival of patients when they were treated with chemotherapy. So it said that biologically we were somewhere on the right track, and those tumors of the ABC type were the real bad apples. We could only cure them in about 40% of the patients with chemotherapy plus rituximab, and then the germinal center tumor is better at 75% cure rate, but of course there's still a lot of people dying of the GCB type of lymphoma, so we care about those as well. Now, another part of the underpinning of this was that these tumors arose from different stages of normal B-cell development. So here is the well-known to immunologists, the Germinal center reaction, in which a B lymphocyte and a T lymphocyte get together with this follicular dendritic cell presenting antigen, and then they develop a specific immune response. The B cells proliferate quite a lot. And from this microenvironment, we believe the GCB type arises from a slightly different cell. Also, Burkitt lymphoma arises from here, another aggressive lymphoma. But the ABCs were different. The ABCs seem to arise from a post-germinal center cell that it's on its way to becoming a plasma cell, but is probably stopped at the plasmablastic stage. So when you then look at the genetics, if you do deep genetics of all these tumors, you find these yin and yang kind of differences in frequencies. So, for example, in the GCB type of lymphoma, you have 45% of them have a translocation of the famous BCL2 oncogene. This occurs never in the ABC type of lymphomas, and we know why, because they express BCL2 transcriptionally, so there's no selective pressure for that. And there are mutations that I'll tell you about in great detail in a few slides that occur only in the ABC, not in the GCB. And then Burkitt has its own set of characteristic abnormalities. So this is telling you this is not one of many ways to slice and dice these tumors with gene expression. These are pathogenetically different. They arise from a different B cell, a subtype, and then maybe need, because of that, a different way to become malignant based on that different epigenetic starting point. Another bit of background before I launch into the data is that this B cell receptor is key to what I'm going to be telling you about. It is arguably the most important receptor in a B lymphocyte. Without it, you can't even make a B lymphocyte. If you don't have it, the B lymphocyte doesn't respond to antigen as it should. You know well about this active type of B cell receptor signaling involving antigen-induced clustering of the B cell receptor and multiple downstream pathways, MAP kinase, PI3 kinase, NFAT, and most characteristically this pro-survival NF-kB pathway. So that we know, but what only the immunologists in the audience know about is another profound signaling mode called tonic B cell receptor signaling, first given to us by Klaus Rajewski. And this does not involve antigen, does not induce clustering of the B cell receptor, seems to only induce the PI3 kinase pathway, not NF-kB, but it's really profound. So if you conditionally delete the B cell receptor from a mature B cell in the mouse, all mature B cells are gone from that mouse in 7 to 14 days. So this is these are two modes, and of course we were interested to see whether they were being utilized in lymphomas. So now our technology. So our technology comes in two flavors in the lab. We have liked for many years genetic screens involving tools such as RNA interference and now CRISPR-Cas9 to look for essential genes, which I'll tell you about. In parallel, we do what everybody's doing, and it's very useful, that's why everybody's doing it, sequencing cancer genomes, and we discover mutations that often make us, give us insights into why particular pathways that we found in the functional screens were so important. So we're sort of converging on these essential cancer pathways. And we're just as enamored as the next guy with CRISPR-Cas9, and we're using the version that cuts the gene that you target, and then if it gets cut enough, the gene becomes inactivated. And it can then be used in a genetic screen such as this. So you take and make a library of vectors that express small guide RNAs that target Cas9 to various different places in the genome. And each one then is targeting Cas9 to a different human gene, different color here. You then infect a cancer cell line, so every cell line gets on average one of these, and therefore is genetically inactivated one human gene. You then impose a selective pressure, and ours is the simplest one you could imagine. Can that cell survive and proliferate for 3 weeks in culture? So this turquoise guide RNA would have hit an essential gene, and because it did, then that cell dropped out of the culture. And you just do next-gen sequencing of the guide RNAs at the end of the experiment, the beginning of the experiment, and voila, you find the essential genes in your cell. So this is a few of our data, not going to show a lot of it, but we're extremely excited about it. And the most exciting thing is this control experiment. Here are quite a large number of control guide RNAs that do not target any human gene. And when we graph them, each dot is a different one of these guide RNAs. And its effect in 3 different cell lines, 2 of the ABC variety in blue and 1 of the GCB in orange. And the 0 means there was no effect, which is remarkable if anybody has used RNA interference, because there were off-target effects out the wazoo. And this didn't prevent us from making progress, but it didn't help much either. So this is a great new day with this CRISPR-Cas9. So what did we learn? So we learned a lot that we knew, and I'll tell you some things that we learned that we didn't know. But first, what we already knew was that the NF-kB pathway was very important in the ABC tumors. So here are 3 components of a IKB kinase, which is the key kinase regulating NF-kB, alpha, beta, and this NEMO subunit. And you can see the dots, the blue dots going down, meaning that this is an essential gene in the ABCs, but nothing is happening in the GCB cell line. And remarkably, A20 is a negative regulator of NF-kB, and when it is deleted, you see the dots are going up. That means it's behaving as a tumor suppressor. It's making these rapidly already growing like weeds cell lines grow faster when deleted. We had never seen this with RNA interference. So then we look at the B-cell receptor pathway, and again, what we knew was that the components between the B-cell receptor and NF-kB were only essential in ABC. So here we have a kinase BTK, very important for a few slides from now, PKC beta, and 2 components of an adapter complex, MALT1 and BCL10, together with CART11, make a critical adapter, signaling adapter complex, all ABC, not GCB. So that was what we knew. Surprise, surprise. So this was a big surprise to us. We had not seen this with RNA interference. When we knocked down the B-cell receptor itself, here showing just the CD79B subunit, or a very proximal kinase SYK, now we did have an effect in this GCB cell line along with the ABC. We think we didn't see this before with RNA interference because you only get a partial loss of the expression with RNA interference. The CRISPR-Cas9 gives you a full knockout. So now I'm going to summarize a whole lot of data from about 12 different cell lines of both ABC and GCB that we've done. And the data is color-coded such that on the left side, the bluer you get, the more essential that is in an ABC-type cell line, average across several. And on the right, the oranger you get, it's the more essential in the GCB. So what you'll see near the top here is the B-cell receptor subunits are all important in both GCB and ABC. And there's some differences, though, in some of the proximal, and in fact, some of the proximal signaling members are more important in the GCB than the ABC. Then you very clearly see that signaling down to the PI3 kinase pathway is important in both of these types. But as we sort of knew and is really borne out here, the NF-kB pathway, all these components, are only essential in the ABC. So we've gotten some clarity around this now to say that the ABC subtype has this particular mode of B-cell receptor signaling, the active form, whereas the GCB has more of this tonic form, and we also found that this was the case in Burkitt lymphoma. And why do we care? Well, simply that this would have different therapeutic strategies. That is, if you have either form, a proximal inhibitor of Syk or Src family kinases right at the B-cell receptor might work. But downstream in the NF-kB pathway, the BTK inhibitors and PKC beta would not work in GCB and would work in ABC. So now pivoting then to trying out some of these concepts in the clinic. So can we target the chronic active B-cell receptor signaling in ABC with a drug, Ibrutinib, that hits Bruton's tyrosine kinase? So first of all, here again is the pathway that is active in ABC from the B-cell receptor through some kinases, BTK, PKC beta, important signaling adapter complex that I mentioned, the CBM complex, and IKB kinase 2 NF-kB. By sequencing genomes from cancers of the ABC variety, we found a bevy of mutations that all, in one way or the other, activate this B-cell receptor signaling pathway. And many of these are exclusive to the ABC type of lymphoma. Just to show you the ones in the B-cell receptor, here the signaling subunits of the B-cell receptor are CD79A and B, and we found every possible amino acid substitution in one particular tyrosine of CD79B. And how that works now is understood that it enhances B-cell receptor signaling in two ways. Prevents the endocytic recycling of the B-cell receptor, so like many receptors upon signaling, you get an endocytosis and an inactivation of the receptor. So with the mutation in the B-cell receptor, this doesn't happen as readily. And then there's a negative feedback loop involving the Lin tyrosine kinase that recruits a phosphatase, and that, for reasons that are not so clear, doesn't work as well if you have this mutant form of the B-cell receptor. But the reason I'm telling you this is that these are sort of backseat driver mutations. And what do I mean? So we know about a driver mutation in cancer. It's one that is responsible. It's in the driver's seat. These mutations, when put in a cell that has no B-cell receptor signaling, do nothing. But if you put them into a cell that has ongoing B-cell receptor signaling, they turn up the amplitude of B-cell receptor signaling. So they're sort of backseat drivers, they're enablers. Okay. So here we were graced with the presence of a drug, Ibrutinib, that was being developed for other reasons to get at autoimmune diseases, but it targets Bruton's tyrosine kinase, and it's a fantastic drug. It's going to be one of the blockbuster drugs, or is one of the blockbuster drugs in cancer, and it covalently inactivates this kinase, so it's very strong, great pharmacodynamics, and we had found in the laboratory it was very lethal for our ABC cell lines. So I'm going to jump through some stuff here. This was then when I had already been working with my friend and colleague Wyndham Wilson very closely, but this was our real moment to test some of the ideas from the laboratory. And very quickly after we met the folks from Pharmacyclics, we did a 10-patient trial, phase 1B trial here in the clinical center. And this was a trial of Ibrutinib in patients that were relapsed and refractory in their disease. And initially we recruited only ABC patients, and here's one of them. So this was patient number 2. She was a 52-year-old female with this ABC lymphoma. Her tumor had a mutation in the B-cell receptor, and like many patients with lymphoma, had some success with some chemotherapy, then a relapse, another chemotherapy, another relapse, and she needed something else. So she came to us onto this clinical trial, just single agent Ibrutinib by mouth, 1 pill a day, no apparent side effects. And here are her tumors in her abdomen in this PET CT scan, and you see before diagnosis, these two gone away at week 8, and now she is with us still, more than 6 years later, and is a happy camper, happy with her service here at the NIH, and we're absolutely thrilled for her. So the first and second and third thought is she? Are you just showing me your exceptional responder, and your results may vary? So of course we put this to a formal test, and this was a 70-patient national trial. looking at all comers of relapsed refractory diffuse lymphoma, used gene expression profiling to subdivide ABC versus GCB, and then predicted, hoped that it would be the ABC tumors that would be responsive. And so we did see that. We had a 37% response rate in ABC and only a 5% response rate in GCB, both complete and partial responses, and some of them were quite good. This is our survival curves. There was a meaningful, statistically significant increase in overall survival of 10.3 months for the ABC versus 3.3 for the GCB. However, although significant, not a home run, I would posit that for these patients largely. There were a few. I mentioned the one lady out past 6 years. We have two other ongoing responses out past 3 years. One gentleman made it to 1.9 years. So I would say we really did move the ball downfield for about 1 in 7 of the patients of the ABC, and we'll get to, at the end of the talk, I hope, how we're going to do better for the majority. But nonetheless, a fairly high response rate in a very bad set of cancers, so we were encouraged. But being the scientists that we all are, we wanted to know why wasn't 100%? You were billing this as the B-cell receptor, the most important thing ever for ABC. Why wasn't it 100% response rate? So could there be something in story in the actual mutations in the particular tumors that helped us? And of course, the first thought was maybe if you have a mutation in the B-cell receptor, then you'll respond more often. And you sort of did, 55% response rate. However, I was taken by the fact that actually 30% of the cases that responded, in fact, the majority of the responders didn't have a mutation in the B-cell receptor. So this then made me think about our data, and especially that this was not what's seen in some cancers, that you must have a mutation to get a response. This didn't seem to play out in our cases. And why not was this idea of a non-genetic form of B-cell receptor signaling, and we sort of already knew this. In our first paper on the subject, we had teamed up here with Sue Pierce, who had been studying B-cell receptor signaling by turf microscopy, and here she imaged some of our ABC cell lines and patient samples and found in red these brilliant dots of the B-cell receptor. So the B-cell receptor was in clusters, and that was not seen in the GCB cell lines and samples. So this was as if, this is exactly what you would see in a normal B-cell exposed to an antigen. So we said, maybe there's an antigen signaling. Well, if that's the case, and these are, our cell lines growing in vitro in culture, what would that antigen be? I guess it would have to be a self-antigen. So we then went out to prove that each of our cell lines relies on interaction between the B-cell receptor and various self-antigens. In one case, a glycoprotein. Another case, an antigen released from dying cells in the culture. And in the cutest example, the B-cell receptor is its own antigen. So there's an idiotope that is right on the variable region that, and by the way, that's what I worked on is for my graduate work, so this is cool to see this. So yes, so in many different ways you have B-cell receptor signaling, and then sometimes you get a mutation in the B-cell receptor that we think augments this process. All right. So now I have to tell you about one other thing that I've neglected on purpose, just to confuse you. But the most important, the most prevalent oncogenic mutation in these lymphomas is in the signaling adapter molecule MYD88. So we discovered that in 39% of these ABC tumors, there were activating mutations that spontaneously turned on NF-kB when they were put in a heterologous cell. And obviously, that might impinge on whether a cell responds to Ibrutinib or not. If it's attacking this arm of the pathway and you have a parallel pathway leading to the same survival outcome. So just to show you these for a second, here is the MYD88 protein sequence, and there's an important domain called the TER domain that interacts with toll-like receptors in innate immune cells. And we had many recurrent mutations, but there was one evolutionary winner that is now called L265P. And this is becoming one of the most important oncogenes in a variety of lymphomas, as I'll tell you about later. And this is biochemically the strongest one of those mutations. So when you put it into cells, here, it spontaneously coordinates a signaling complex involving two kinases, IRAK1, in which IRAK1 is heavily phosphorylated and active with IRAK4 to signal downstream. As you see, don't see that with wild type. So it's a gain of function for the NF-kB pathway. So how did it play out in our trial? It was sort of a wash, okay? So nothing going on there, let's move on, except that we noticed there were 5 tumors that had both B-cell receptor and MYD88 mutations, and 4 out of those 5 patients responded. And 0 out of 7 patients that had only the MYD88 mutations responded. So that was a significant difference. And it did jive with what we were finding in the laboratory. This is a response to Ibrutinib, a killing assay for Ibrutinib. And these are a variety of cell lines. In blue, the ABC, the 3 that are obviously having effect have both mutation in the B-cell receptor and MYD88. These other ABC lines that aren't touched by Ibrutinib only have a MYD88 mutation, and the GCBs are not touched. So that seemed to look like hyperaddiction to B-cell receptor signaling in those double mutant cells. Genetically, we also knew that if you survey a large number of ABC tumors, there are more that have both mutations than you would predict by chance by the prevalence of either mutation by itself. So 10% of the ABC tumors had both mutations. So that was genetic evidence that these were cooperating pathways. And then a little bit of functional evidence was that if you inhibit MYD88 and then you read out B-cell receptor signaling by phosphotyrosine or phosphosarc, in every other lane is a MYD88 inhibitor, and there's less signaling from the B-cell receptor somehow. At this point, we didn't know how, and you can say the same thing if you knock down MYD88. Now, in all walks of life, in science also, sometimes seeing is believing, and we love this assay called the proximity ligation assay that allows us to see signaling inside cells. And so it's a very simple thing that asks whether 2 proteins are near each other that you think are involved in signaling. And that is, you have to have a different species antibody to each protein. You then come in with a secondary anti-species specific antibody that has oligonucleotides coupled to it. And if these 2 proteins were close enough together, a rolling circle amplification of DNA is initiated. And then you visualize that with fluorescent nucleotides, and you get a fluorescent ball exactly where this protein-protein interaction is. So here is a proximity ligation assay between a B-cell receptor CD79A subunit and MYD88. And you may see, it's a little hard, that there are red dots. This is obviously the blue nucleus. These dots are not on the cell membrane where a self-respecting B-cell receptor ought to be. These dots are in the cytoplasm, so that was a surprise. And if you, as a little control, knock down MYD88 with Cas9 CRISPR technology, you lose this signal. So it's a lot of controls that I'm not going to show you. So how do we understand this cytoplasmic co-localization of MYD88 and the B-cell receptor? So we knew this. This is from our CRISPR screens. CRISPR screens to the rescue. So here we knew MYD88 was essential, and here is one ABC cell line. Various guide RNAs for MYD88 are depleted. Here is then, you can look, because we do whole genome CRISPR experiments, what's happening with the toll-like receptors that are some of the receptors that use MYD88. And pretty much nothing except toll-like receptor 9. So clearly, and this was a new finding. We didn't know this before we did the CRISPR screens. Now, this was important to us because TLR9, unlike some of the other toll-like receptors is not on the plasma membrane, but rather in a cytoplasmic vesicle, an endolysosome. And it gets there by a unique pathway involving some chaperones in the ER Golgi, UNC93B1 and Canopy 3. And obviously our thought then was maybe this is where the B-cell receptor and MYD88 were. And we had some genetic evidence from our screens that here are sort of a lot of data, sorry, but here are the data for MYD88, TLR9, UNC93B, Canopy 3 across a large number of cell lines. And the only cell lines that depend on all of these are the double mutant cell lines. So this whole pathway of TLR9 and its biogenesis is important in these double mutant ABC lymphomas. So then here's a proximity ligation assay between IgM and TLR9 in the same bright cytoplasmic dots. And if you knock down with CRISPR-Cas9 the B-cell receptor or TOL9, you lose these dots. So this is a specificity control. So I'm going to show you the model first, and I'm not going to have time to show you all the data, but some of the data that supports this. And we have fondly termed this the BCR-MYD88 super pathway in ABC lymphomas. But it takes as a starting point that every receptor recycles into the endosome, but we think that some of the receptor then gets shuttled into this endolysosomal compartment where it meets up with TLR9, and there we think is where all the NF-kB signaling, and there is where MYD88 is, there is where the card11 MALT1 BCL10 complex is, and there is where IKB kinase is active. That's our model. We're sticking to it, and I'll tell you why. So here is, first of all, a proximity ligation assay between two components of that signaling adapter. Fine, you get nice spots. But the genetic experiment is here. If I knock down the B-cell receptor, we lose those spots. This we knew. But we did not know that if you knock down toll-like receptor 9 or MYD88, you also lose those spots. So that means there's a signaling crosstalk between the MYD88 TLR9 pathway and this thing that had previously been thought to be devoted to the B-cell receptor pathway. Now we asked, could we show actual signaling at this cytoplasmic location? How would you do that? So we thought, well, what if you took the substrate of IKB kinase and asked whether it, in its active phosphorylated version, is there in these dots? So we simply had an antibody against the substrate phosphorylated IKB alpha and did a PLA with TLR9 and IgM, got these spots. You notice that they're seen in the ABC cell line, not the GCB. But then the functional experiment is if you then do a knockdown of either the B-cell receptor or TLR9 or MYD88, you lose that association of that substrate at that cytoplasmic location. So that's sort of our evidence that that is where active NF-kB signaling is taking place. All right, so now take a breath, but I'm going to tell you one other story that will then was logically leading from this insight in the lab back into a clinical trial. And in this, we were paying attention to the literature, and we're finding that there were mutations in MYD88 in other lymphoma types, other people's work. And so we started wondering whether other lymphoma types might use this B-cell receptor MYD88 super pathway, as we're euphemistically calling it. And it's pretty impressive, the frequency of MYD88 mutations. So in our ABC lymphomas, that 1-point mutation, L265P, is in 29% of cases. This is in garden variety nodal lymphomas. But if you have these extranodal lymphomas, either of the skin, often on the leg, or in the testes, or in the brain, here the rate of prevalence of this point mutation is 60% roughly. So these seem to be enriched, if anything, for maybe this kind of signaling. And even more so, in a couple of studies, had started looking at the B-cell receptor mutations that we had described and found that, as I mentioned, well, this is our work, in ABC lymphomas, here sort of re-upped, 11% have both mutations, whereas in primary central nervous system lymphoma, 37% of those tumors have both mutations, and overall, more than 3/4 of these tumors have one or the other of these mutations. So Wyndham and I started looking at this data and really came to the conclusion that we had to do a clinical trial with Ibrutinib in this primary central nervous system lymphoma. Now, to set that up a little bit, it's not a great disease. It's not a great anatomical place to have a rapidly growing aggressive lymphoma. It's a lymphoma that has a ABC phenotype by gene expression profiling. I showed you the genetic relationship with ABC as well. Here's the survival with chemotherapy. What's sort of not shown here is these patients get a lot of brain radiotherapy as well, and that has very bad sequelae. So we need a lot of help on this disease. So we decided to do a phase 1 trial of Ibrutinib plus an optimized chemotherapy I'll tell you about in a second. And we were looking for overall response rate and progression-free survival. So we devised this special regimen that we really favor, where the targeted agent, Ibrutinib in this case, is given by itself for 2 weeks. The patients can tolerate this, and that gives you the ability to see the activity of that drug by itself before you mix it with chemotherapy when you really don't know what's going on. But we do the chemotherapy because we want to help these patients, right? So, and the idea is we give the Ibrutinib with the chemotherapy not because we think Ibrutinib is going to cure them, but that the Ibrutinib, by decreasing the apoptotic threshold, might make these tumors more susceptible to the chemotherapy. A lot of data behind that. And Wyndham, and I will not go through this, in all of his years and years of drug development in lymphoma, came up with an optimized regimen, completely changing some of the agents that are normally given to patients of this type to optimize for brain penetration of active lymphoma agents. And I think that was also very important. So first, a patient. This was a 67-year-old. She had a presentation with difficulty speaking and walking and had a cerebellar mass. She had a good run with chemotherapy, a 3-year complete response. She had some radiation therapy as well. And then she had a recurrent disease, inability to speak, and that's when she came to us. And so within about the first week or a few days even, she started talking to her family, so that was a moment. And then here's her tumor going away partially, a good partial remission. Remind you that it's only 2 weeks of therapy with this drug. So how have we done, and oh, by the way, and she's still now in complete response following the chemotherapy part, taking no treatment 2 years out. So far, so good. These are early data. And so we are just as cautious as we can with early data, but this is a really good response rate. So here are 18 patients, and 17 out of 18 showed a decrease in their tumor. 16 out of 18 met objective PR response rate to Ibrutinib, so a much higher rate of response than we had seen in the nodal form of ABC lymphoma. When we combined it with chemotherapy, 87% went into a complete response, and then many of these are ongoing with the arrows, and it's early days. We're still following these patients, but the refractory patients who had been unresponsive to their previous therapy or their tumors were unresponsive, had a median progression-free survival at this point of 8.2 months. So that's hopeful, and we're now starting to ramp up for a phase 2 trial to take this farther. In the last 2 minutes, I will tell you where we're going in the future really quickly, and leave some time for questions if there are any. So obviously, I may have shown you that there were active responses, but like in any targeted therapy, and you've all been paying attention to these wonderful new drugs that come through for cancers of this type and the other, and as soon as they're announced to be wonderful, the next announcement is we're having relapses and resistance mechanisms to these targeted therapies. So this is the beast of cancer that we're trying to defeat. So one way to get around this is to work out mechanism-based combination therapies, and I want to emphasize that these are not empirical combinations as earlier drugs were developed because that was the only thing you could do. Drug combinations for chemo worked out fine, but there were no mechanisms or fewer mechanisms thought about there. So now this is a little busy slide, but I wanted to show you in all its glory what we think of as the inner workings of an ABC cell line and in a cancer in general. And what's shown here is some things you've seen before: the B cell receptor at the surface, the NF-kB pathway. But I'm also showing you that these lymphomas, at least in our cell line models, have a minimum of 4 different survival pathways. They have the PI3 kinase pathway that's triggered directly from the chronic active B cell receptor signaling. The NF-kB pathway has as its downstream targets cytokines, IL-6 and 10, that trigger a pro-survival JAK-STAT signaling. Then we have a variety of BCL2 family members. BCL2 itself expressed at high levels and sometimes amplified genomically, MCL1 expressed, and then NF-kB causes the expression of BCL-XL and A1, another set of survival anti-apoptotic pathways. So you look at this, and then you say, you've been foolish. You're going to try to conquer this mess of signaling with one agent targeting only one of those 4 survival pathways. So we've been going at this issue hard and thinking that we have to find rational combinations that hit these other survival pathways. One of them, we've been fortunate enough to work with Craig Thomas and NCATS quite closely, and he has a nice combinatorial drug screening platform where you can do thousands and thousands of drug-drug interactions, and that's been very illuminating. And then sometimes we have come up with interactions just by staring at this wiring diagram and making some educated guesses. But in the end, here's where we are. There are clinically available agents, all in blue, that all either kill our lymphoma cell lines directly, and many of them synergize very profoundly with Ibrutinib in killing these ABC cell lines. So we have no shortage of things to think about. The problem is somewhat different. So just to give you, before telling you the problem, just show you we have moved forward with one of these combinations, Ibrutinib plus lenalidomide. By the way, this was definitely not as hard as an act of Congress, perhaps, but it was difficult to get 2 different drug companies to work together on a combination trial. And we showed them the science over and over and over again, and they finally bought it. They finally bought that the science made sense and allowed us and sponsored this trial, Celgene and Pharmacyclics. And one of the bits of science was that when we did a xenograft model of ABC lymphomas, and we sort of used not a fully active dose of either Ibrutinib or lenalidomide, we could clearly see that the 2 together were quite synergistic in controlling tumors. And so this study is ongoing, so I have no real results, but an anecdote that we've seen a lot of responses. This is a person with a recurrent relapse. It doesn't look quite so nasty just there, but it's a bad one. This was a recurrent disease, and this can kill you. And it's gone now. These are just discolorations in the skin. So we will read out the final results of this trial late or in middle of this year. So we'll see. We know it's an active combination, and equally important in targeted combination therapy, safely administered. So this is important. You can do a number to patients if you aren't careful by mixing these inappropriately. So where our thought process is going, and I don't think that everybody in cancer thinks exactly this way, but it is what it seems obvious to me, is that every cancer, I believe, has multiple survival pathways. It doesn't need just one. It probably needed different ones at different stages of tumor evolution. And that if we imagine that each pathway has several nodes in it that could be targeted with different available drugs, so there's 4 drugs. I haven't told you much about the inevitable resistance mechanisms that come up. Let's say there's just one resistance mechanism and it has 2 nodes. There's 2 more drugs you have to consider. And in a simple example, there would be the 8 different clinical trials that would mix and match the drugs shown on here to try to overcome all of these mechanisms. So that's not going to work. There's too few patients that are going on to clinical trial, and I don't have enough patients, to use a different form of that. It's too slow. So I think this is a great opportunity for scientists to think and add value to clinical work where we have to nominate the best trials, best combinations based on mechanisms that we understand, and hopefully predict that they will also be safe and try a fewer number of those. So I'm going to stop here, and I got actually one more sort of acknowledgments after these traditional acknowledgments. The traditional acknowledgments are that Jim Phelan, a postdoc in the lab, led our CRISPR screens. He deserves a lot of credit. Ryan Young led our work on the crosstalk of B-cell receptor signaling. He's a staff scientist in the lab. All of our structural genomics has been done by Roland Schmitz, a postdoc, and a staff scientist, David Wang, for informatics. The IRF4 work with lenalidomide, I didn't tell you about IRF4, but that was the work of a postdoc, Yibin Yang, together with my longtime 20-year anniversary staff scientist, Art Schaefer. And in big capital letters, I told you about Wyndham Wilson's contribution and his team. And collaborators in laboratory pathology, Stefania Petaluga, in other parts of the NCI, George Wright. I mentioned Craig Thomas at NCATS, and external collaborators. So for fun, I would tell you who my mentors are. Now, when I got started, the word mentor had not entered the lexicon of science. You sort of just did your work and you learned from your environment, but nobody sort of said, you know, Lou, here's what you should do. But they sort of hinted at what I should do, and I was attuned to these things. So here is my PhD advisor, Walter Gerhardt at the University of Pennsylvania. He was the epitome of a laboratory scientist, immunologist. And here was his thought for me: you just have to love pipetting. He honestly would say this to me. He said you would have to come into the lab and not think about making a single discovery, just enjoy the process of doing experiments. And I think that's so important. And I you know, it's a real chill job in that sense. You just come in and you get to run your own lab and your own experiments and make your own work for yourself, and it's a really lovely way to think about your career in science. Now, I had another mentor at Penn, and that was Martin Weigert. Very, very famous immunologist, discovered somatic hypermutation of immunoglobulin genes, and very influential on me in a quite different way. So his approach to me was to get within about 2 inches of my face and say, that's the stupidest thing I've ever heard. And this was very formative, and you would think it would be terrible, but it was just fantastic, because then I'd say, but no, and you know, I'd start to struggle and answer him back, and that was really, really important, and sort of toughened me up a little bit, shall we say, as a scientist, which is very important. Now, really important in my life, probably the most formative moment in my scientific career was being a postdoc in David Baltimore's lab at the White N Institute. And it's very hard to express exactly how he influences you so profoundly, but I thought this might get it. So what he would say from time to time, sparingly, would be, "That's amusing." And this was the highest form of praise. This is, "You did not bore me. You did not waste my time. This is worth considering. You are worth considering as a person, and your experiment is worth considering." So this just set for me an incredibly high standard for what you were supposed to achieve in science. And so I've kept that with me ever since. So then, sitting in the front row is another extremely important mentor of mine, Tom Waldman, who recruited me here, and I don't have time to say the very fateful way that all happened on a beautiful island in the Mediterranean, but that's another story. But nonetheless, so informative, so influential in so many ways. I'll have to explain this one. I'd like you to attend clinical rounds. Okay, so he said many good things. This is not the only good thing, but this was important when I think back on it, because in fact, he had Friday rounds that were just standard, what you'd get in medical schools, or they were seeing the patients on the ward, and here's what happened in the past week. And, I was a basic scientist at that point, and I was running about as fast as I could away from clinical medicine. I had gone to David Baltimore's, it just was, but, I said, well, he's my boss, I'll come on Friday and soldier on. But it did imbue in me this importance that he feels in his research that I now feel very strongly. To have this relationship between what you do in the laboratory and whether you can help patients. And so that I credit Tom for. Another famous face, you're all familiar with our friend Rick Klausner, and he was really helpful in jump-starting my career in genomics. And he was just became the NCI director when I was ramping up. And we were together, he said, "Lou, you're just not thinking big enough." Now, does that sound like Rick? I think so. And what he meant by this was that we had found some good results, early results on, maybe 20 or 30 tumors. And what he said is, we're going to get everybody in the world together in one room that studies lymphoma, and we're going to see who has lymphomas and who has clinical data, and we're going to profile all of them, and it's going to be great. And so this was Rick to a T, and I have embraced that kind of gung-ho mentality. That's very, very important, I think, for success. And then my final mentor that I'll call out specifically is Harold Varmus. So the next important NCI director for me, in many ways, had a lot of this David Baltimore phenotype of really making you want to up your game to the highest possible level. That was very important. But here was his special thing. He said, "Lou, I may need a little help from you." And this was a very understated way, like in him saying, "I want you to do this other big job for me that's directing all genomics for the Cancer Institute, and don't stop working in your lab either." So this is what that very naive phrase meant. And I actually am very grateful that he gave me this opportunity, and I haven't talked about this today, to direct large projects. What I usually say is, you take our budgets in the intramural program and add a 0 or 2. That is what our budgets are on the other side that we do on these national large projects like the TCGA. And that's really fun to think about. It's a different mode of thinking than we do in the laboratory, but it has its own rewards. So thank you very much.
Thanks, Lou. And I would warrant that there are a number of people on this slide and lots of people in this room who are influenced in a similar way. And certainly my career was by a number of these people. So you'll take questions, I guess.
Yes.
Yes.
Awesome lecture, very inspiring. You mentioned Ibrutinib is completely safe, which was surprising to me since we know mutations in BTK result in immunodeficiency. Can you?
Yeah, I oversold that a bit. So it's pretty darn safe as cancer drugs go. So the surprise, though, is there is no obvious decrement in normal B cell function. The normal B cell numbers are untouched. The serum immunoglobulins are just down a little, IgG and IgM. And in most patients, there is not a heightened susceptibility to infection. What we have found recently, though, is that if you have probably other immune suppressants on board, such as steroids, then you can get in trouble with BTK. And we have worked with Michael Leonidas at NIAID to show that BTK function in innate immune cells, most likely, is important for controlling certain fungi. So this is not, it's not a nothing, but these patients, most of them, thousands of them, are taking Ibrutinib without any side effects. So that is a true statement, and that's why this is a special kind of drug.
And that leads me to my second question. What about combinations? IR4 is very important for the immune system as well. When you target both of these pathways, when you have some problems.
Right, that's exactly it. And I was getting to that. I didn't get at it very deeply, but we're mucking with essential pathways, in our case, immune cells, but even more generally with other cancer therapies. And if you hit two things the wrong way, there's going to be some normal cell in the body that won't like that much. So it's really all about therapeutic window and what you can get away with. So and I won't regale you with some really, really bad outcomes in clinical trials where people have had two different target agents at the same time. So we are well aware of that. But as I mentioned, we're doing an Ibrutinib plus lenalidomide trial, and it does not appear that we are, creating havoc. It's tolerated.
To follow up on that, there is evidence in the literature that lenalidomide and Ibrutinib affect immune cells other than B cells. So my question is, can you comment on these effects and also the potential for combination therapy with small molecules like lenalidomide and Ibrutinib with immunotherapies?
Great question. I think that's an outstanding thought that there will be a synergy between immune therapies and targeted therapies for the malignant cell. In fact, in our study that I showed you, the phase 2 study, when the patients were here at the clinical center, we were able to get in some of them biopsies on treatment. So pre-treatment and 3 days on treatment with Ibrutinib. And the signature by gene expression profiling that showed up was CD8 T cells in the responding lymph nodes, not in the ones that didn't respond. There was too small numbers, so we didn't put it in the paper. It's anecdotal, needs follow-up, but it's along the lines you're mentioning. Over here first, yes. And then Larry.
Just very briefly, when you showed the first slide of anatomic pathology and saying it's precision medicine, there was an intermediate step of immunoperoxidase. Those of us who trained in the 80s in anatomic pathology remember the days of S100, which was essentially all we had. And you've essentially chosen the hardest area of pathology, hematopathology. Back then we had what was called the working formulation, which nobody understood what it meant anyways. And I think the implications for precision medicine is also precision pathology, because this has enabled people to do tumor of unknown origin, which we used to not do, and to figure out if metastases are really from the primary that you're seeing, or you have 2 simultaneous tumors, or 3, and believe it or not, in the literature, those used to be very big topics. Apparently, they're no longer important now that you have genetic looking at tumors, and you can actually say, hey, this is from that tumor, or this is from that.
Right. So I think that the important point that I take from your question is that pathologists should embrace these new technologies, and they have always embraced technologies. They embraced this early molecular study, as you mentioned. Immunostaining is a molecular test of the tumors. We're just doing tests of hundreds and thousands of genes at the time. I think that the science of diagnosing disease has traditionally been in the laboratory of pathology, or in the hands of pathologists. And if pathologists want to stay part of this solution in precision medicine, they've got to embrace these techniques, I think.
Thank you.
Thanks, Lou. Do you have any more to say about the germinal center type and approaches there?
Yeah, thanks. Absolutely. So there, I mentioned that it was the B-cell receptor tonic signaling, and their PI3 kinase seems to be important. And in fact, all the PI3 kinase mutations involving PTEN and all that are all clustered in the germinal center. So the genetics also is pointing directly there. Likewise, we in Jagan and Mupadi here now recruited, and Jason's sister showed there was a separate pathway involving the S1PR2 receptor that is a negative influence on PI3 kinase pathway, and that's recurrently deleted. So everything is converging on PI3 kinase. But then, on the other hand, you've got BCL2 translocated in 45% of the cases, and other BCL2 family members, especially MC1. So the concept that I put forward, maybe not as complicated as ABC, maybe we just don't know enough, but at least there are two different survival pathways. So I think great combos would be involving the ABT199 BCL2 inhibitor and some PI3 kinase inhibitor in GCB to make progress. Okay.
Okay. So let's thank Lou for a terrific MITRE lecture.